So Mesoplasma florum is a molecule. It's part of the Spiroplasma group. So it's a close cousin of Mycoplasma mycoides and Mycoplasma capricolum. I've been studying Mesoplasma florum in my lab for about 10 years. And the main reason why I started working on this organism is first because I was introduced to this bacterium by Tom Knight during my postdoc at MIT. And I thought it was a super cool organism. And also it's non-pathogenic, so it was very easy to work in BSL-1 level labs, and it's a fast-growing bacterium. So it doubles every 31 to 33 minutes, depending on the temperature and the medium. So that's as fast as E. coli at the same temperature. So that allows us to do very rapid experiments. My goal today is to give you a brief overview of what my lab has been doing with Mesoplasma florum for the past 10 years or so, and then to give you a preview of what will happen in the coming months and years, so that you can see the differences and the similarities between the research done on GCVI, SYN3, and Mesoplasma florum. So a few years ago, when I started my lab, the first thing that we did was to do comparative genomics, because we were interested in knowing what was the core genome and how many genes were conserved between species. We also looked at the pangenome, we looked at synteny, and we discovered that the Mesoplasma florum genomes are pretty much all equivalent. They have some slight differences, but they pretty much look alike. And using transposon mutagenesis, we used a library that Tom Knight made at MIT, and we classified the genes based on their essentiality in the lab using the transposon, and also with the conservation of the gene in the core genome between all the Mesoplasma florums. So that allowed us to do different scenarios of what could be a minimal genome or a reduced genome. These are gross approximations, and they're probably not that relevant. And the estimation varied between 657 kb to 290, depending on the criteria that we used. So we decided that we wanted to do something a little bit more sophisticated before starting to minimize the genome. So the first thing that we wanted to do was to make sure that we could actually do some genetic engineering. So we developed genetic tools such as integrative plasmids, antibiotic selection markers, promoters that we knew worked really well, and we integrated yeast genetic elements in Amphorum, and that allowed the whole genome cloning in yeast. And later, with the help of our wonderful collaborators in Bordeaux, Carol Lartigue and her group, we were able to do genome transplantation. So transplanting Mesoplasma florum genome from yeast into Mycoplasma capricolum to generate transplants. And that was the limit, and I think it's still the limit of the phylogenetic limit of genome transplantation, where Mesoplasma florum is right here and capricolum is right here. So I don't think that transplantation beyond that point worked, although I might learn some new things today. So knowing that we were able to do transplantation, then we did a deep cellular characterization. So we wanted to know as much as possible about this cell. So we looked at the genomic features. We also mapped the transcription units, the promoters, the terminators. We measured the physical properties, which was much harder than we anticipated. So we now have a good idea of the diameter, the volume, the mass of the cell, the biomass composition. We also looked at growth parameters, and then we estimated the number of molecules per cell for all proteins and RNAs. So that was published last year, and we then decided to use as much of this information as possible to build a metabolic model for Amphorum, and this was published a few months ago. So this is the metabolic map for Mesoplasma. It's pretty similar to other mycoplasmas. There's a reduced metabolism inside of the cell, and there are a lot of transport reactions. And using this model, we were able to predict different phenotypes. And one thing that we also did was to use the min genome algorithm And that's something that's a tool that uses transposon mutagenesis plus metabolic models to predict blocks of genes that could be deleted. And in this case, we impose different constraints on the growth rate. So trying to keep the cell growing as fast as possible or trying to be more tolerant. So this gave different scenarios that were slightly different. And the genome that we predict would be growing would be 562 kb with 535 protein coding genes. And one thing that is also interesting is represented in this figure here. So the blue circle here are the genes present in SYN1. The green circle in the middle here are the genes present in SYN3. And the red circle here are the genes present in Mesoplasma florum. So if we predict a minimal genome and we compare it to SYN3 and SYN1, we find that some genes are shared, and they're right here. So the majority of the genes are shared, but there are some particularities. So there are genes that are only present in florum. There are genes that are in SYN3 that are not conserved in florum. And that leads us to think that there's probably more than one minimal genome composition. And although Mesoplasma and Mycoplasma are close cousins, they might not use the same solutions to get to a minimal version. And that brings me to pretty much where we are right now. So having these papers published, we wanted to have a better transposon mutagenesis data because the density was a bit low with what we had from Tom Knight. So we started working on the defined growth medium and high density transposon mutagenesis. So the first thing that we started to do after the work on the metabolic model was to try to have a completely defined medium, which contained purified albumin to supply the lipids. And then we also played with peptones and peptides to supply amino acids and sugar as carbon sources. And what I really want to show you here is that with that defined medium, we're able to get very good growth densities that are as good as medium with serum, right here represented in blue. And now we can change the carbon sources and we can get good growth rates, high densities. So using this medium right now, we're doing high density transposon mutagenesis. So what I mean by high density, we did 80 transformation that yields between 10,000 and 15,000 mutants per transformation. So we have about 1 million mutants. And then we grow this pool of mutants in different conditions and different carbon sources. And we do multiple passages, and then we sequence the transposon insertion sites. At the same time, we're also growing cultures without the transposon inserted into them, and we do RNA-seq. And we hope with this, with the TN-seq and the RNA-seq, to be able to define some groups of genes that are co-regulated and to associate them with a most likely function in the cell. So that's on the part of understanding. In the part of engineering, we've been working with serine integrase. So they're unidirectional integrase that are derived from bacteriophages. And we've been testing a few of them. I'm showing you BXB1 and FeC31. So if we transform 1 microgram of a plasmid, the plasmid will get into the cell. We have recombination mediated by the serine recombinase here on both sides of a gene of interest. And we insert at a site-specific location in the chromosome. So we can easily get a few thousand mutants or clones every time we do this. And we've been testing different things with this platform, which I believe is probably similar to SYN3B. One of the things that we're currently doing is testing different randomized promoter libraries in order to get promoters of different strength and to build inducible systems. We have a lot of different candidates right now. We're confirming everything, and we'd like to write a paper about this pretty soon. Another method that we are trying with serine integrase is to divide the genome into different sectors, and each sector is flanked by 2 different ATTB sites with a central dinucleotide that is different. So that allows to use one serine recombinase with different landing pads or recombination sites. And we're working right now on one section, the one right here between 40 and 80 kb in the Amphorum genome. And we're building two different versions at the moment. One that is recoded, so we're changing the GC percentage and the codon usage, and another one that is recoded and refactored. So refactored means that we're separating every feature on the DNA. So if we have a ribosome binding site within a gene, we're separating them. We're separating the RBS from the first coding region and isolating it. So that way we could later move the pieces of the genome more easily, we think. We're also planning a reduced version of this section, and we hope to have it in the coming weeks or months. And the goal, the engineering goal of Mesoplasma florum for us is to have a toy example, a toy genome with which we can play and make things modular, try synthetic promoters, add enzymes, use unnatural amino acids. We recently inserted the arginine energy genes in Amphorum to see if arginine could supply the energy for growth or complement the energy that we get from sugar. And we hope that in the coming years we can go way beyond the phylogeny that we see right now and have completely artificial bacteria. So in conclusion, my group is doing systems biology of Amphorum to understand the cell behavior and the gene functions. We're also doing synthetic biology to engineer genomes, but also to learn about genome design rules. And I hope that I can convince you that by comparing between Mesoplasma florum and GCVI SYN3, we can better understand the differences and the similarities between minimal genomes, because I think that there are different versions and different possibilities. And by comparing, you learn a lot more. So with this, I'd like to thank the lab members that did most of this work. They're listed here, and the funding agencies. Thank you.
Very nice, Sébastien. It almost makes me think that, you know, if we had been smarter, we would have chosen Mesoplasma florum many years ago, although I don't know if we would have ever gotten transplantation to work, since it was considerably less efficient than the mycoides to capricolum transplantation efficiency.
One thing that I did not mention because of the time is that we're trying to transplant from Mesoplasma florum to another Mesoplasma florum. And we just recently identified one strain, the W17, which is super good at receiving DNA. So we have about 1 million clones per microgram of DNA, and we usually get about 1,000 for the L1 strain. So there might be some hope for transplantation between Mesoplasma florum strains.
Very good. Anyone else? Questions?
Very nice talk, Sébastien. You mentioned about the serine integrases. I think it's a very clever idea also to evaluate some possibilities. And let me put my video. I'm Elibio. Sorry about that. I mentioned my name. And very nice talk. What about, have you thought about using all the serine integrases in order to generate a circuit and able to switch on and off some of your genes? It might help to give us some clue about the importance and function. Please comment on that.
So far, we have tested 4 different serine integrase. That's something that we thought about. We could engineer recombination on it or inversions of certain elements. Right now, we got good yields only with BXB1 and PC31. So we did not explore that further, but that's definitely something that would be interesting in the future. For example, in terms of inducing or having very tight on-off switch for some circuits.
Thank you. Sounds great.
Here is,
Go ahead, please.
sorry, John. Here's Bogumił Karaś. Thank you, Sebastian, for this kind of great update. Always very excited. So I have a question, because this is, as John said, really great platform in addition to the Mycoplasma platform. Now, do you think there is still need to develop another platform that will be with standard genetic code? Because Mesoplasma, as far as I know, right, it is not standard, just like Mycoplasma, Mycoides or Capricolum, or the synthetic cells. So is there a goal to turn one of those cells into standard genetic code, or is there a need to develop another Mycoplasma or another molecule into a similar system with standard genetic code?
So you're right, the Mesoplasma florum use code 4, so same as Mycoplasmas. So some genes that we have in E. coli will not necessarily work in Mesoplasma florum, or we have to recode them. And what we've been doing for the moment is code them with a compromised codon table so that they work both in E. coli and Mesoplasma florum. I'm not sure it's essential to engineer a standard genetic code. There are some advantages, but you can also argue that if you have a different genetic code in terms of biocontainment, that's also a barrier to horizontal gene transfer and a good thing in terms of biosecurity. So that's not something that we're planning for the moment, but it would definitely help moving things around between E. coli and Mesoplasma florum or yeast.
Okay.
It's also worth noting, if you remember Keone Gandel's talk last week, that there is, through the BioBricks Foundation, the Free Genes Program, a Mesoplasma florum data set of genes using the standard genetic code.